So, I think I'm online now. Hello, everyone, and yeah, welcome to another Saturday live stream. I'm happy that you could join again. I'm just doing a little bit of housekeeping again here. Yes, I can actually see myself as well online. I think the sound should also work. Yeah, so here we are again. Let me quickly check again. Yeah, there are now 29 people watching. It's nice. Okay, good. It seems to be working. I'm always a little bit nervous at the beginning because sometimes there are some technical issues as well. So, yes, today I'll be doing yeah, an interesting starch reaction. I see interesting because I'm also going, yeah, I'm just going to explain to you later all of the details. I just tried out something today, which I would like to share with you, something that I've not seen before like that. So there is going to be a little bit of a novelty here as well. So I'm going to just say hello all around the world here. Okay, hello from UK, from the UK again. Okay, from Vietnam. Okay, Vietnam again, from Finland, from the United States, the Netherlands, from Scotland, Georgia. Sound is good. Thank you very much. Okay, and yeah, so and that's a very sunburnt banana. Sure. Hello from Canada, Hamburg, in Germany, Romania. Okay, great. Okay, so last week. Right at the end of the, I think of the live stream, somebody requested that I put some starch under the microscope, starch grains, and indeed they are quite nice to look at, especially if you're using polarized light microscopy for that. And I decided to thank you for the recommendations, and I'm going to show you some starch grains, including an interesting starch reaction. I mean, many of you already know that iodine and starch react, but look what I got here. You can make the whole thing a little bit more interesting by adding a little bit of vitamin C. What in the world does this have to do with that? Okay, yeah, I'll explain. And if you're new to this format here, there is a chat, a live chat on the side here, and please feel free to ask questions. I will try to answer them, and usually after about 1 hour, I will just then not do any practical work anymore, but then simply only answer questions. But please, if you want to have, if you want me to respond, then please write at Microbehunter there, because then it's much easier for me to find the questions, okay? And every now and then I'm just going to interrupt myself a little bit and simply say hello from the to the Philippines, Poland, the United States, okay? Yeah, yeah. A carpet beetle is on the desk. Oh, you got a carpet beetle on the desk. Yeah, I think carpet beetles are also known as museum beetles, and these museum beetles, if I'm correct, they have very, very interesting hair. Yeah, is yeah. So if it's actually the same one here, so basically a little bit of an explanation here. What in the world happened to this banana here? Well, actually, I kept this banana in the refrigerator for yeah, quite long, and usually bananas turn brown sooner or later. It's still fairly solid, so I'm going to show you then a little bit some starch grains from banana, and of course from a potato. And what I'm going to do first, I also have a little bit, yeah, a little plant here, and I also discovered a few starch grains here. But actually, the really interesting thing is the iodine reaction. And I know that some of you, or many of you, are already familiar with the method. Yeah, bear with me. I'm just going to show it to you anyway. And then, yeah, I'm going to do the following, and that's actually the nice and the new thing, is I'm going to reverse the iodine reaction with vitamin C. And this can also be observed, of course, under the microscope. Okay, so what I'm going to do first is, yeah, I'm going to quickly show you some commercial slides. Okay, and yeah, that's here. That's a commercial, yeah, microscope slide here. Let me switch off the arrow again here. And you see, yeah, from a potato, and you see that those yeah, pink things here. And unfortunately, it doesn't say what stain it used. You see that these pink structures here, round things, these are the starch grains from a potato. You can also, of course, see this in the cells here, the cell walls. So I think this is actually quite a nice, yeah, nice specimen that we've got here. But unfortunately, I don't know what stain they've used. Yeah, so I'll be using in any case iodine. I don't think that this is iodine because the starch grains stain pink. Iodine makes them actually stain dark blue, black. Yeah, so that's one thing, and another one here, and this is this one over here. This is an unstained one. By the way, I'm looking at bright field right now, right, and that is the cross section of a wheat grain. Okay, and you can see that those tiny little dots that you see. These are all individual starch grains. Yeah, and of course. Yeah, seeds do contain a lot of starch because they need that as a food source for the young plant to grow. And those starch grains, I'm going to now try to also show you in a potato and also in a banana, and I'm going to stain them. Then, yeah, I'm just going to experiment around with them a little bit. Okay, so I'm going to interrupt myself again here. The larvae have the interesting here, the beetle itself doesn't. Okay, But where there are beetles, maybe I'll soon find larvae somewhere. They here are barbed along the entire length. Yes, that's correct. As a matter of fact, they have quite long and interesting hair. Okay, so another comment here. Are you sure the banana is not moldy? I'm quite sure it's not moldy. It's been brown. I've never, this doesn't matter. It is just basically the banana simply has changed its color, but it's definitely not moldy because then there would be mold. On the surface. Yeah, and as a matter of fact, I'm quite sure that it can still be eaten. So we're going to do that later, and I'm going to just show you another preparation for the potato first. You see, the potato is also not the best one here, because we need a lot of starch grains, and we're going to look at them under the microscope, and then we're going to stain them, and I'm going to show you a couple of what I consider at least interesting features here. So my recommendation is as follows. I mean, there are multiple ways how you can do that. I always do it like this. I'm going to make a fresh cut here. Yeah, so that's much nicer. You know what? I'm going to lower this here a little bit here. And I'm now going to get out the starch grains. And in order to do this, I add a little bit of water. Okay. You take a knife, and blunt side is fine. And that's all you do, is you just scratch the surface and you kind of dip it back in here. Okay. Yeah, and okay, and that's pretty much it. I mean, there's already more than enough. But you know what? I'm just going to cut off another slice here. Yeah, because when there's a fresh surface, then the cells have broken open here, and this is the place where we can get out more of the starch. Yeah, so we can keep on doing this a little bit. Okay, there's even some pulp here. Yeah. So, and I think this might actually be enough. Okay, and I just put the potato away for right now. So, and then the next thing that you do is you kind of try to do it like this, and you try to collect whatever you've got down here. Okay, so that is essentially where we've got a lot of starch, and I'm just going to put this one directly under the microscope simply to show you some unstained starch grains. And my recommendation is if you have a regular bright field microscope, get yourself some polarizing filters and put it between two polarizing filters because it really looks nice as well. So let's just put it on a slide here. Occasionally I'm reading again your comments here. I'm just going to skip ahead. I'm just because there's so many comments, which is really great, but I'm just going to read the ones that are directly addressed to me. The name of the how do you call this? We call them a spider plant here. I don't know what it's called. Yeah. Yes. I'm skipping over the off-topic questions, and I'll answer them later. Yeah. Do other fruits and vegetables have starch too? Of course. Yeah. Of course. Yes. A lot. Beans, corn, whatever. Right. So we'll put a little bit on here. Cover glass goes on top, and I'll just show this to you first. Unstained starch, and then we're going to do a little experiment because So I have to always, you see, it's always a little bit of a difficulty getting the, so. Because starch is denser than water, it will actually collect here on the edge. And we're just gonna have a look here under the microscope. And yeah, I need to switch, of course, scope view. Oh my gosh, what happened now? I lost the camera. Is this really the case? If this is the case, then I'm really sad. What happened here? Oh my gosh. I don't have a camera. Oh. There's some USB problem then. What in the world? Let me just quickly check again. No, it's actually there. It's the stage. Uh-oh, folks, if this is the case, then I don't know what to do. No, I actually lost the camera. Why is that? And if I close OBS Studio, then essentially, oh my gosh, then I'm in trouble. Just give me a second. I don't know how to, yeah, HDMI stage. So that's the wrong one over here. Yeah, look, I've lost the camera. Oh my gosh. You know what? I'm gonna try. You see, folks, if I can't get this to work, then I have to basically start a completely new stream under a new link. Then I have to restart the computer. It used to work. It just worked, right? So what's going on now? That's not the one here. Oh my gosh, I don't know what to do now. Deactivate it, and then let's try to activate the camera again. I'm panicking. Try to switching to another scene. Yeah, I'll do that now. I'm going to switch to the stereo scene. That's the stereo microscope view. So I'm going to switch now back to the scope view again. I mean, the arrow is here. The stage I can turn on and off. My face is here, but this one over here. No, it's... how am I going to do this now? I'm a little bit worried. Yeah, I guess I have to unplug the USB, but then I'm kind of worried that it's going to be... Okay, just give me a second, okay? So, let me see. This one over here, it's this here. So I don't know if this is unplugged it, plugged it in again. I heard the sound. We're back. I cannot believe it. Ooh, okay. I think we're back. Okay, I think we're back. Here we go. Here we go. Okay. This was a stress now. Okay, thanks for your patience. Okay. Yes. Yeah, that's indeed. With as Jesse is saying, there is indeed an issue probably with the hub, but normally it works because there are too many cameras connected. But usually, what I do is I always switch the cameras off when I change scenes. But for whatever reason, it's yeah. Okay. Yeah, this was a little bit of a panic. Yeah, I hope that this is gonna work now. But at least, yeah, here we go. We can continue now. So what we see over here is, and I guess I'm just going to leave it like this now. What we see over here is, and this is all bright field, by the way. This is all bright field, by the way. We see over here, of course, the starch grains. And if you look very carefully, you're able to see that there are rings on here. So I'm going to go up to 60. Yeah, and those rings are actually the individual layers as the starch grain is deposited. Now these starch grains are crystalline, so this means they're extremely hard and difficult to digest. And when you heat them up, then they become larger and softer and amorphous. But they basically are pretty difficult to digest when raw. No? So good. Yeah, good. Just some comments that I'm reading here. So what I would like to do now is I would like to now take it a step further, and I'm going to now add a little bit of iodine, because that's actually the thing where we, yeah, that we have to. Now I do have to switch scenes again. So let me quickly switch back to the desk. The desk is okay. Back to the scope. Back to the scope. Microscope also still works. Okay, good. Yeah. So back to the desk. Here we go. So I'm now going. So I've got over here iodine Lugol's solution. You got to be careful with that thing a little bit because it's a very strong stain, and I once spilled a little bit on my floor. Impossible to get it out because it actually goes into the wood. And in a concentrated form, you also have to be careful that you don't spill it on any technical or on your microscope or so, because it's kind of corrosive. In that sense, it's not so good. So, but what we do now is we just add a little drop or two, yeah, here, and you might actually see that, yeah, you can see that it's starting. Some of the, yeah, it's starting to turn. This is black thing here, right? Yeah, and these are the starch grains. Yeah, so what I'm doing right now is I'm just going to flush them down here, and I'm going to wash it because I want to remove the iodine again, right? And then I'm going to add water to make sure that the iodine is gone. Yeah. So how does the plant or seed break the starch down using cellular respiration? It looks hard to break those hard starch grains. Yeah, it uses amylase, as a matter of fact. So to break it down, that's actually also in the brewing process. Yeah, when you use wheat or to make alcoholic beverages, then the plant has to slowly start to germinate in order to produce the enzymes to break down the starch to make sugar. Yeah? But look at this, how black it is now, right? Let me also all these things here. Let me remove. Yeah, I've got a little my little jar here as a yeah. So. Also the bigger particles remove. And I'm just going to add a little bit of water here, and I'm just going to rinse and wash everything because I want to remove the excess iodine. Yeah. So, and about the USB thing, I have had multiple issues with it already because sometimes it changes the ports. So this means it kind of confuses the cameras as well. So I have to get this somehow sorted out. I've got I bought myself separate, of course, separate USB adapters, of course, because I've got, I don't know, 6 cameras or so connected here. That's, of course, a lot. You have to, when you connect them, you have to make sure that the cameras are connected to different ports and different controllers so that one controller does not have too many cameras connected. So all of these things have to be taken a little bit into consideration here. So we're almost done here. Yeah, but that's gonna be fine. So this is fairly concentrated and yeah, plenty of starch. And I'm going to now make a slide here yet again, and then yeah. So still so far nothing really totally unusual. Up, you don't see it because here we go. Yeah. So, and of course a cover glass. I lost my cover glasses. Yeah, okay. So let me quickly read a couple of comments here again. Yeah. So how does the plant seed break the starch down using cellular? Well, let's put it this way. It doesn't break it down because of cellular respiration. The plant produces the enzymes that break down the starch to make maltose, and then it uses the sugar. It uses the sugar for cellular respiration. Yeah. So, yep. So we go here. We put this on here, and let's have a. There's too much liquid on here. Because for the next experiment, we really need very, very, very little liquid. Okay, so all of this excess has to be actually, all of this excess liquid has to actually go away. Okay, all of this excess liquid has to go away. Okay. Should be really very, very little water. The reason is because I will be then adding vitamin C to the starch grains. Okay, so yeah, let's have a look. So now let's see if it works. If it still works. Ah, I heard that it disconnected when it switched over. Ah, it didn't. It made the sound. Why did it do that? Why does it do this? Why does it disconnect? Let me do the following. I don't know why. That's a different scene. I don't know why. Why does it do that? It's kind of really strange. Okay, I'm going to try to unplug it again and plug it back in again. I'm in panic mode now, actually. Yeah. Yeah. I keep on losing the USB port for whatever reason. Yeah. So, does this mean that every time when I switch back to the desktop view, I have to go beneath my table to? I don't know. So that is basically, these are the starch grains. Yeah, as expected. Now they look quite nice and black. Okay. So, and however, what I wanted to show you is something slightly different. Is I would like to maybe disconnect the objective view to reduce the load of the other cameras. I could try to do that. Yeah. What I'm gonna do is the following. Actually, I've said it like this: that if I change, then it should be. Yeah. I'm just gonna play it safe now. I'm just going to remove this view here. So you're not able to see this here. So this is gonna go away now. Yeah. Okay. So that's one thing. Then number two, I'm just going to play it safe. I'm going to do the following. Look, I'm just gonna move this here now over so that it looks a little bit better. So, okay, I guess we're just gonna be happy like this, yeah? So, yeah, I have, the crazy number of cameras attached. That's, yeah, a little bit unfortunate. So, and now what I would like to do now is the following. I'm gonna risk it again. I have to go back to the desk view, unfortunately. or I'm going to try to disconnect also the other cameras that I have. I'm just stereo, so that's the stereo camera. I'm going to disconnect that here as well. Stereo stage, I'm going to deactivate that as well. And I'm also going to deactivate the stereo camera. I'm just going to deactivate pretty much everything I've got. And let's hope that the scope view, yeah, this, okay, so this one still works, and I'm able to switch back and forth now. Okay, so I just activated a couple of cameras, and then maybe this is going to, yeah. So number one. Number two, now I would like to show you something here, which I have, which I knew that this is possible, but I've never seen this under the microscope before. The thing is the following. If you now add vitamin C, then the vitamin C is going to react with the iodine, and it's going to convert the iodine to an ionic form, and therefore it should actually lose the color again. So what we're going to be doing is, by adding vitamin C, I want to reverse the starch reaction. Okay, and this actually method is used to actually determine how much vitamin C there can be in fruits, for example, or in vegetables, whatever, in orange juice. If you want to detect the amount of vitamin C, you can actually do that. It's a so-called a titration experiment. However, what I'm going to do is now, by adding the vitamin C to the black starch grains, I hope to be able to reverse this reaction. Actually, yes, not only hope, I know it works because I tried it before. And this is simply something I would like to show you. Okay, so let's do the following. I'm just going to take a little bit of this vitamin C. You see, this is here pure vitamin C. These are not vitamin C tablets or anything, which contain a lot of other things as well. But this is actually pure vitamin C powder. And actually, if you want to eat this stuff, you shouldn't eat more than just a knife tip. Yeah, we add a little bit of water. Okay, and we're just gonna dissolve this here as much as possible. Yeah. And let me see. Yeah, I think that should be enough. And I'm now going to do the following. Now you don't have the camera, so you don't see what I'm doing. I'm just using my pipette, and I'm going to add a small amount of this vitamin C solution to the cover glass. Okay, and then this should actually, a little bit here, a little bit of vitamin C. So I'm going to switch back to the scope. Let's see if it works. Does it work? Yes, it still works. Good. Okay. And now I'm going to add this vitamin C solution. And of course, this is going to flush away the starch grains, but we just want to focus on a few of those in any case. Yeah. So it's going to start flushing away the starch grains. Let's move there a little bit further. And what you notice, look what happens to the starch grains. Look, especially the one on the bottom, in the center, from the outside in. Do you see this? What happens? Yeah, they start to turn clear again from the outside inwards. Yeah. And the black color starts to disappear again, and we've restored the original color. Yeah. I think this looks kind of nice here because you can actually see how there's this ring of clear starch, and then in the center it's still black. Yeah. And the reason is because the vitamin C reacts with the iodine and removes the iodine, so to say. I mean, it doesn't remove it; it converts I to I minus. Yeah, so it reduces the iodine. So it's a redox reaction, oxidation reduction reaction. Yeah. So can starch be heat fixed? I don't know. Can starch be heat fixed? I don't know. Normally, heat fixing works because proteins cross-link with the glass. Okay. So I don't know about. We could try it. Okay. And over here you see, yeah, it has not reached it yet. I'm right now right pretty much in the center of the cover glass. Yeah, so yeah, or the vitamin C is already used up. This could also be. Yeah. So let's go down with the magnification here, so we can actually see this. This actually looks pretty pretty here. You see over here there is still clear, and here, yeah, there's still black. So this is the place where it did not reach it. But on the outside here, yeah. Now this one a nice one here. Look in the center. Let's make it a little bit brighter. So that's kind of the thing that I kind of wanted to show you. And yeah, by essentially determining how much you basically, what you do is you add orange juice or something like this, whatever contains vitamin C, and then if you measure the amount of orange juice that you have to add, then you can calculate also the concentration of vitamin C that was present. Yeah. So I just wanted to show this to you because I thought it was kind of neat. Yeah. If we dry the slide, will it then form crystals? I wonder. Well, let's put it this way. Let's put it this way. That's an interesting one. The vitamin C certainly will form crystals. Yeah. I mean, you know what we could try? I'm just going to leave this here. I mean, I do have my coffee warmer still, yeah. So it would be an interesting thing, a combination of vitamin C crystals and starch grains, yeah. So, yeah. So I think it's kind of interesting, yeah. You can actually see here how the vitamin C diffuses, either into the starch grain or that the iodine diffuses out and reacts with the vitamin C. Yeah. I just wanted to tell you another thing, and I was unfortunately not able to repeat that experiment under the microscope because we humans, of course, also have amylase. And what you can do is, and I got it to work once a couple of years ago, made even a video, I remember, where when you add now saliva to starch grains, but those starch grains have to be heated and boiled, then you're also going to see that how it digests away those starch grains, and it will also start to lose the black color. Yeah, so that is also something that I once tried, but I was not able to reproduce it, and even today I was not able to quite reproduce it, unfortunately. Yeah. So you know what I'm going to do is I'm just going to leave that the slide, and maybe by the end of the video, maybe in an hour or so, maybe it will have formed a few crystals. We're going to see, yeah, because vitamin C likes to form crystals, but at the same time evaporation is a little bit slow because there's this cover glass on top. Yeah, you know what? I'm just going to leave it as it is, and otherwise I'm just going to try to connect my coffee warmer, and then maybe we can speed up the process a little bit. Yeah. So that's basically the thing that I wanted to show you. I'm going to show you now, just for reference purposes, what happens when we heat the starch grains. For that, I need again my little burner here. So let's go back to the desk. Okay, and I just wanted to show you what happens when this is heated up. I need a slide because, interestingly, we also lose the color when we heat up. Yeah, the slide. So here is the burner. But why do you need to heat to let amylase break down the starch? You don't need the heat. The reason why I'm going to need heat now is because it will change the structure of the starch grain. Okay, so that's the thing that, and I just wanted to show you a little bit how the structure of the starch grain changes after boiling. And that's the reason why we have to boil our potatoes so that we can eat them. This is because the starch becomes amorphous. So, but I want to show you now the following, because just watch the black color here, because this is actually quite interesting as well. Okay, look what happens to the black color. Okay, let's give it a little bit of time. Yeah, do you see? It's starting to lose color. Okay, yeah, here, yeah. So what I'm going to do now is I'm just going to add a little bit more water. Yeah, it's heated again. So I'm boiling now the starch. You see, and it's completely, yeah. So it completely lost the color. So what I'm going to do now is after when cooling, I'm going to add a little bit more iodine, and then we're going to have a look at the banana, because the banana is also quite nice to look at, and we're going to convert everything into black color. Okay, and I'm going to turn off the fire, and I'm going to wash everything again. Okay, so this means you see all of these, I don't know if you're able to see this. I'm going to remove all of this excess. Where's my trash? Here it is. Yeah. You see now, how am I going to do that? And yeah, I'm just going to put this okay, so I'm just going to put a cover glass over this here again. Let's have a look. Okay, so I'm again, every time when I switch scenes, I'm nervous. Every time when we're still good. So this goes on here. So let's have a look now. And this is yeah, basically how it now looks, and it looks pretty ugly, right? So what has happened is let's go down with the magnification yet further. What has happened is that the starch grains are basically completely boiled apart. Obviously, yeah, you still have a starch reaction, but they've completely lost their crystalline structure as expected. That's the reason why when you boil, yeah, potatoes, of course, they become soft. Yeah, they're still, yeah, still some semi-boiled. And let's give it also a try. Let's also add a little bit yeah, of vitamin C. Let's see how this works. I've not tried this yet with the amorphous, yeah. Ah, look, it also works. Actually looks also pretty cool. I'm going down with the magnification to see this better. Look. Yeah, how the black color disappears. Yeah, so yeah, the vitamin C completely removes the iodine again. Well, quite nice to look at. I wonder where these bubbles come from. The bubbles seem to yeah, form and then disappear. Interesting. Yeah, yeah. But I think you get the idea that I guess these bubbles must have been caught a bit beneath the starch. Yeah. Yeah. So you see actually how. Yeah, so that is this traditional, how do you say, this vitamin C iodine reaction, which essentially is used to detect the concentration or the amount of vitamin C, but right now we're just looking at it under the microscope, so to say, what's actually happening. Yeah. Yeah, and I think now it stops because either the whole thing has already been used up, or I'm just going to add a little bit more of this vitamin C solution. Yeah, and it continues a little bit. Yeah, yeah. So a couple of things here again. When heating it up, then you know that the starch essentially starts to become amorphous. It loses the crystalline structure. Also becomes easier digestible this way. Yeah, and unfortunately, I was not able to make it work with my own saliva. Yeah, even though this worked already in the past, I've done some experiments in the past, but for whatever reason, it didn't work. Yeah, because there's of course lots of amylase, that's the enzyme, in my saliva which digests the starch. Yeah. Okay, so what time is it now? Okay, what I'm gonna do now is let's move on to the banana. Okay, so this was the one from before, the one with the vitamin C, and here now I remove this again, and now let's have a look at the banana. Desk view. Yes. So, and here the starch grains are also quite nicely visible. And how am I going to open this here? I think I need to clean the knife a little bit, otherwise I'm going to cross-contaminate it with the starch grains from the potato, which I don't want, obviously. So, in case, because it's been mentioned, is the banana moldy or not? No, it shouldn't be moldy. Let's open it up. Look, it's perfectly fine, right? Just to show you that it's perfectly fine. Yeah, look. It's just ripe, but it didn't even turn brown on the inside. Yeah. Yeah, so perfectly okay. So in order to, let's do the same thing as before. I'm just going to show you the original one, and then I'm going to show you, the one stained with starch. So soft bananas are quite easy to prepare. You take a little bit of the banana, put it on here, and the next part is the fun one. You take, of course, a cover glass. I clean it a little bit, and then make sure that the surface is hard, like a plate. You take a tissue paper, fold the tissue paper, you carefully press it, put it over, and you just press vertically and hope that the slide doesn't break. Okay, until you have a very thin film. You see, it's already so thin that, maybe you can even try it even more by compressing it like this. No horizontal movement. Okay, and then let's hope that we're able to find a few starch grains here. Okay, so again, microscope view, and then, yes, here we go. Okay, I'm going to put on the condenser again. Let's go up with so everything is in bright field. Yeah, if you want, you can also use DIC. DIC looks more colorful. Yeah, so that's basically with a little bit, some colors here. And all of these things here that you see this? Yeah, these are all, of course, starch grains. Let's put the filter away so that you actually see it better. Yeah. So you see that plenty of starch grains. That's why banana gives you so much energy, because you can also make banana chips, for example, and these spiral-shaped structures, of course, you already know. That's xylem. These are basically vessels that transport water. They are spiral-shaped. You see all over here, all of these are the starch grains, of course. So over here, these lines that you see here, these are essentially the cell walls of the cells. And of course now the integrity of the long xylem over here for transporting water, right? And of course then in between everywhere starch grains, which we basically want to stain now again. Yeah, simply to make them also better visible. But you know what? Why not use a little bit of DIC just for the fun of it? Yeah, and yeah, so we can have a few colors. Yeah, a little bit colorful. Yeah. Let's go up. I don't know if I've never really checked if we're also able to see the layers of the starch. Yeah, so we have to make it a little bit bright. DIC really even eats away a lot of light. Yeah, but maybe slightly, or like this, it's too bright. Yeah, that's no regular bright field again. Yeah. You see the streaming of the liquid, yeah? Because I compressed it so much, everything was under tension, and now it's as the cover glass tries to loosen itself a little bit, you have all of the streaming, yeah, of the liquid here. Why can't we see phloem? Well, chances are pretty let's put it this way: the xylem is easy to see because of the spiral shape, because these are the so-called the strengthening rings. Of course, phloem will also be there, but it's difficult to identify as such. Yeah, very often xylem and phloem are next to each other, so I can imagine that maybe. Now I'm gonna guess. The spiral ones, it's xylem. Maybe those long cells next to it, maybe that's the phloem. The xylem transports water up towards the plant, and the phloem transports substances away downwards. Yeah. You can even see over here that inside the xylem, yeah, there is even an air bubble now, yeah, because these are very long, thin tubes. Yeah. Everything is always so beautiful blue. That's because of my DIC. The DIC is a specific technique that uses polarized light. Yeah, yeah, that's a regular bright field. If you also want to get colors, you can always play around with color filters, of course. Yeah, but I usually, yeah, especially when making YouTube videos and so on, sometimes it's simply more interesting to look at if there is color present. So, so that's basically number one. Okay, now let's do a little bit of staining again. So I need again a Petri dish. I'm always not really become nervous now to yeah, every time when I change. And we now do the following. We again take a little bit of this banana. That's a huge one. Yeah. So. And let's add a little bit of iodine directly to it. And yeah, how am I going to do that? I do not want to touch, you see, in order to mix it, metal, nah, probably not, because it's corrosive, the iodine, right? So maybe I'm just going to use some disposable toothpicks to kind of maybe mix this in a little bit better. Yeah. And of course, because now the starch grains are not directly exposed, they are inside cells, therefore the iodine is not able to reach the cells quite as easily. And for this reason, it might take a little bit longer for the iodine to diffuse into the cells. Yeah. So, I think, but that's, I would say probably already enough. It's way too much in any case. We're going to do the following. Okay, let's just do it the easy way. Let's just flood everything. Okay, and we take again now we can pick a small one over here. Let's just say this one. Difficult to grab. So here we go. Yeah, cover glass goes on top. You know what? I want to rinse it in any case yet another time, because I just really, I really want to make sure that most of the iodine is actually gone. So that's good enough. Cover glass on top. Cover glass on top. And just like before, we're gonna give it a nice squeeze. I put this away here. Let me put it somewhere where I'm not spilling anything. And let's give it another squeeze. Yeah, here we go. Let's have a look. Okay, this is how it looks like. You can already see that something's there. And scope view. I wonder why it's always flashing. That's kind of weird. And here we go. Look, isn't this nice? Now we can actually see the individual cells also much better. I think this is pretty cool, right? Let's just stay here. Okay, so yeah, obviously over here this is a cell, a banana cell. Now also much better visible, probably because of the iodine, and of course inside here we've got the starch grains. Yeah? So you see, this is actually a quite a nice, simple experiment. Well, experiment is a kind of demonstration, so to say. Yeah. So I'm just occasionally reading. Have you finished your slide vacuum chamber? Not yet. What vacuum should it have to make the bubbles disappear? Well, honestly, what you can try to do is the following. I don't have it here now, but there is a very, because you're asking, maybe I should answer this later on when I'm talking about the off-topic questions, but these syringes, right? And you can buy very large ones, extremely large ones, which are not used for injecting, but actually for feeding. For people who can't eat, you kind of have a tube through your nose into your stomach, and you can put some food, some liquid food into that. And some of these syringes are so large that you could actually fit a whole slide in there. So if you want to kind of take away the bubbles from a slide, all you do is you kind of close the opening here, and then you pull, and then this creates a vacuum, and this would also remove then the bubbles. Just some side, so this, let's go down a little bit with the magnification to get a better overview of the overall pattern here. So it's down. So the arrow, the bubbles of course are visible, but I think this is actually very nice because now we can actually see the individual cells much better, right? And plenty of starch is present, and those starch grains look quite a bit different. Yeah. So I do have two more examples here that I would like to show you. One of them is basically directly from the packaged cornstarch. Let's have a look at the cornstarch. Okay. I'll remove the slides first. Let's put them away. The cornstarch is, I guess we can just look at it directly. I need a clean pipette, a little bit of water. I don't know if you're able to see. And we're gonna add a bit of this starch. So this was basically sold of course, it's sold in supermarkets for making food. If you want to make some kind of a sauce or cream, and you need to thicken it a little bit. Yeah, let's see how this works. Yeah, this is probably way too much liquid again. Okay, so let's have a look here. This is now cornstarch, scope view. Yes. And to make it a bit brighter again. Okay, they look a little bit more as if they have a little bit slightly sharper corners. So that is basically commercial cornstarch. Let's add a little color again here. I don't know. I need to flip this over here. Yeah. a little more light, and you can play around like this. Yeah, yeah. So that's another one. Okay. So some comments here. Why banana starch different shape compared to the potato counterpart? Well, I don't know. Let's put it this way: on the very basic level, starch is basically long chains of so-called alpha glucose connected together. Why it is different, I don't know. Because let's put it this way: if you want to have an evolutionary explanation, then because it works. Yeah, and all that living things care about is that they're able to survive, and evidently different starch grains basically work work equally well. Yeah? Thank you for the food syringe tip. Ideal for people with less resources or less tech savvy. Yes, I've got this food syringe, and you just have to close off the tip, and then I was able to actually fit in a slide. But you got to get really big ones to get enough vacuum, right? Or you can try to connect the food syringe to another plastic container with a tube, and then you can also try to suck out some of the air. Yeah? So bananas lose starch as they ripen. A ripe banana contains 1% starch, a green banana 80%. Some argue that it's healthier to eat green bananas instead of ripe ones. What's your opinion? I don't think that this is a question of starch alone, right? I mean, just because something contains more starch does not automatically make it healthier or less healthy, I would say. I don't know if there are no other substances inside a green banana which maybe might not be so good. So I probably, when eating bananas or fruits, would probably not base my decision on the amount of starch that's present, right? Because in a ripe banana, what you've got is in a ripe banana you've got you've got more sugar, because more of the starch probably was broken down. That's why ripe bananas taste sweeter, right? It's also I would say maybe a question of digestibility, but I would not. My opinion is that these things probably don't depend on the concentration of starch that's present. Yeah, that's what I would probably say. Yeah. Generally, health issues are quite complicated anyway, because sometimes there is not a direct correlation. Yeah? I mean, how often do I eat a banana? Maybe once every couple of days. That's probably not going to make a huge difference to my health, I would probably say, because there might be other factors that play a stronger role. Yeah? So, what else is there? So we've got the banana, we've got the cornstarch, and there's another thing I just wanted to show you very briefly. No, not show you. There's one thing I wanted to try out, which I didn't try out yet, and I would like to do the following. Let's see if I'm able to get that's a little bit risky. That's not a stereo view, because I do need the stereo microscope now. Okay. Let's do the following. Let's try to activate this in the hope. I wonder if the stereoscope works. What I would like to do is I would like to make a very thin cross section of a potato. I've always scratched it, but I would like to make a cross section of the potato, and hoping to be able to see the starch grains inside the potato cells. Okay, so this is a little bit the new thing that I want to try. So, and for this reason, I need to make a very thin cut, under the stereo microscope, and this means that I'm going to be disappearing a little bit for a couple of seconds, and you will only be able to see that what I'm doing under the stereo microscope. So stereo, let's see if this actually still works. Yes, it works. Let's remove the arrow. And I'm going to try to move the microphone a little bit closer, but I don't know if you're able to hear me quite well. But I'm going to now disappear, and I'm going to now move over to the stereo microscope, and then you're going to see what I'm doing. I need a very sharp razor blade. Okay, let's give it a try. No, you cannot see it because the magnification is too. Okay. So I have to focus. Which direction do I focus? Here we go. Okay. It is pretty huge. Okay. So I want to make now very thin slices, and I don't know if this I'm able to get this to work, or maybe this is actually too thick. This is too thick. I'm going to let me slice this across like this. Okay. So let's see if this works. French fries. So, okay, let's give it a try. I don't know if here it is. Here it is. Okay. So I'm gonna try to put this under the microscope. Here are the comments and the questions. Thank you again. So, and let's see if this actually works. Yes, I actually found out that making very thin slices under the stereo microscope is actually sometimes even a little bit easier than using a microtome, ironically. So let's have a look. Well, I could use a microtome. I have used a microtome, but actually it's faster also for the live stream, for the sake of the live stream. It's a little bit faster simply cutting it under the stereo microscope. So let's see if the desk works again. Yes, here we go. I need some kind of tweezers. And I don't know if this is actually gonna work, but let's give it a try. I need always be careful with those razor blades. Let's move it away. I lost my pipette. No, here it is. Water goes on top here, and I hope to be able to see the starch grains inside the cells, and I don't know if this works or if the cut is not a little too thick. Yeah, we're gonna see. Microscope, let's see if the microscope still works. Yes, it works. We're closely getting there. Okay, here there's some starch grains that came out. Looks actually quite pretty. Ooh, way too thick. Way too thick. Maybe a little bit. Maybe a little bit, I don't know. Yeah. Kind of, maybe. Let's go up. I think the cut was still a little bit too thick. Let's go down. Yeah, I should have. Maybe something like a microtome is indeed better. Yeah, but the idea here is to show a little bit how the starch grains are actually inside the cells. Here you might be able to see it a little bit. Yeah, there's this cell wall around it, and then you've got the starch grains inside here. Yeah. So, and those here on the side, they kind of were able to escape because the cells were cut open. Yeah, so this is definitely the cross section is definitely too thick. Yeah, but I think you get the idea a little bit of what I was what I intended to show you. Yeah. So okay. Yeah, at least I'm relieved that the camera still works. I don't know, maybe I'm gonna try to get myself a new USB 3 or USB card, one with several controllers on them, because as I mentioned already before, sometimes you've got one USB controller and then you've got several ports. Oh, I'm getting too technical now. And you always have to make sure that the load is somewhat distributed. Yeah, no, this was not a very how do you say? Yeah, this one here maybe where the grains are free, but I wanted to actually show you a little bit how it looks like, but I think I made the cut a little bit too thick. Yeah. So let's see. Is there anything? So I looked at the starch grains on the potato from the last one. I prepared something over here. Last one. I'm just going to remove this here again. So we looked at potato starch. We looked at corn starch. We looked at, but this one starch that we did not look at yet. And here it is. Don't shake it too much. This here is rice wash water. Some months ago I made a video even, and we're just going to have a look at the starch from rice. I let it stand. This one here is pretty fresh. I just made it before the live stream, but one of them I let stand for, I don't know, a couple of days, and then it was full of bacteria, of course. So I need my pipette again. Yeah. I'm just reading PCI with SATA power and using the headers from the motherboard for higher data throughput. Generally the best hubs available, often cheaper too, but depends on the quality of the provider. Yeah, PCI with SATA power. So do you actually mean to say that there are PCI boards? I mean, some boards that where you have to connect extra power to it, maybe. Yeah. So like some graphics card, of course, they also need to connect extra power supply. So maybe I have to also find one. Yeah. Actually makes the micro manipulator. Yeah. Yes, there's this comment over here. Last week I was playing around a little bit with a highly improvised micro manipulator. I think calling it micro manipulator is a little bit of too much. Yeah, but so here we go. Let's have now a look at the starch grains from rice. It always flashes, interestingly. I don't know why. These things here that you see here, that's something from the cover glass. But indeed, look. And the ones from rice, just like before, look highly irregular. And what I think, is that, because they don't look particularly nice, really. And if you add now iodine, they do stain black. But what I think is that maybe because of the rice, the processing of the rice and the removal of the how do you call this, the seed coat of the rice, maybe this kind of scratches the surface, and therefore you do not get very nice starch grains. Yeah. But these are highly irregular, so I think that these, the fact that they look so irregular is actually due to the processing of the rice, because the surface was kind of abrasion and it was kind of rubbing off, and therefore it could be maybe that the original structure of the starch grains was kind of destroyed. Because if you remember at the beginning of the live stream, I showed you the cross section of a wheat grain, and the starch grains, they were quite nice, but those here from rice water, not really. Yeah. So this is actually something that I've already seen before already. Yeah. And I think that this is indeed because of the processing. The rice has been processed. And to make a control that this is indeed starch, we're going to do the following. With a vitamin C slide. Where did I put it? Oh my gosh, I don't even remember where I put it. Maybe this one is the one. So we can try to stain it. Was this the one? Let me see. I should have labeled it. No, that's not the one. I'm not. Am I? If not, then I misplaced it. I misplaced the slide. Or is it this? Was it this one over here? in my perfect chaos. No, that was the banana. You know what I'm gonna do now? Honestly, it's gotta be, folks. I'm going to do now the following. Yeah, because I also want to know. I'm going to now do the following. I'm going to take again some of the potato starch, and I'm going to now, just a second, get my coffee warmer. This one over here. So, and I'm going to dry the slide using the coffee warmer. Because at the beginning I promised that I want to see how it looks like to have vitamin C and the potato starch grains next to each other. And that's actually something I've never seen before, and that's why we're just gonna do it the first time together here on this live stream. So, I've got, so why not? I'm just gonna take this here. So I'm going to take a slide. Here is a slide. I'm going to move this up here. I'm going to go to the desk again. Today is a little bit, as you can see, today is a little bit chaotic, everything. So, we move this away. And I've got, over here, I've got the banana. Where is my starch? Is over here. Is from the potato from before, and I'm now going to indeed. I want to now mix the iodine with the iodine. So it already dried up. So that is, oh, you couldn't see it. So over here, that's again the stained potato starch. So, and now I want to add the vitamin C. Where's the vitamin C? Here is my vitamin C. Okay. And we should be able to see that it will start to disappear again. What we can do is, let's try, just because it was so fun, let's try it again under the microscope. Yeah, I know I've got the questions, your questions there, but I already did answer so many of them, or a few of them, but I want to give this now a little bit more priority. So, and then let's add again the vitamin C to the whole thing. Let me see, scope view. Yes, here we go again. Let's remove the thing. Yeah. So, and now let's add a little bit of the vitamin C. But I'm going to go down with the magnification quite a bit so that we get a better overview. So, we remove the arrow. And we're going to add now the vitamin C. Let's... see. Ooh, it looks actually also quite nice. Here we are. Higher magnification. Here we go again. Look how it starts, how they start to become clear again from the outside inwards. Yeah. Here it's not reached it yet. How does this look with DIC? Yeah. Also fine. So, okay, and now what I'm going to do now that I added some of the vitamin C, let's, I'm going to now put it on the coffee warmer, and I'm just going to dry it very quickly so that we see how vitamin C, crystalline vitamin C, and those starch grains together look like on a slide. It's a little bit of a, why not? Why not have a little bit of fun? Yeah, this is actually quite interesting. Look, yeah, you can actually see quite nicely over here and over here. Yeah, so the vitamin C, as it approached, it completely has removed. And I wonder whether the iodine itself inhibits or has any kind of effect on the crystallization process. I wonder. I don't know. I really don't know. So, what makes the starch grains so shiny? I'm using now polarized light. And polarized light actually, because the starch grains themselves are crystalline, so you can actually see that the starch grains, yeah, because they're crystalline, they also have a polarizing effect on the light. Yeah. And this is the kind of, and also now with DIC, and this is kind of why they look so shiny. If you just want to see it the normal way, then this would be the way that you see the normal bright field. Yeah. Here again. See that they're kind of losing the color from the outside inwards. So, okay, over here is my, how do you call this, the coffee warmer. And where is the desk view? Here is the coffee warmer. And I'm going to put the slide on the coffee warmer. Then we're going to see if this actually, this actually has an effect or not. Oh, thank you to Jesse. Thank you very much. Okay, I've got he sent me an email with a link to a PCI hub. Thank you very much. No, because I tell you honestly, just to tell you a little bit, I've been really struggling. I mean, just last September, I bought myself this new PC, new computer, simply just because I'm doing so much video and live streaming and so on. Before that, it was actually quite even worse. So it got better, but what I've seen is that sometimes when I start the computer and also OBS Studio, that sometimes the cameras are switched around, right? So I downloaded a program where I can actually assign different names to the cameras, and it still didn't work perfectly. So this is, yeah. And indeed, in case you are also doing something with cameras or live streaming, any one of you, you got to keep the USB cables short. Yeah, so that's also can sometimes be a source of a problem. Yeah. So if you use two polarizers and cross them on starch, I would see pretty sparkly things more. Yes, let's put it this way. I cannot do that, unfortunately, because my microscope here is from the configuration. But what I've got here is the following. Now, these are from old photo cameras, but you can also get plastic filters. And it's difficult to see, but when you rotate them, and what you do is you put the slide with the let's use the commercial one here, in between here, right? You cannot see it now because there's no light coming from the bottom, but when you go into cross polarizing position, then the starch grains are going to shine brightly, so they're going to look quite nicely, so. Yes, 1 to 2 meters max cable length. I had honestly before that, and which really didn't work. I think at 4 or 5 meters cable length and put together over several extensions. Of course, this is not going to work properly. I'm talking about the USB. Yeah. So the situation improved now for me because now I only have fairly short cables and of high quality. Yeah, but that's essentially what you have to do. Is you have to kind of cross it, and then you might be able to. You know what? Let's give it another try. Just a second. Let me see if I'm able to get that to work. Not this one here, this one here. Let's see the stereo. Yes, stereo works. Look, what I'm doing right now is I'm going to put this here on. So I've got now down here, I've got this is the polarizing filter, okay? As you can see, it's the polarizing filter. Then I put the sample. This was the one from the beginning of the live stream, to focus it a little bit. And then I put the other polarizer on top of it, and then I rotate the whole thing. Yeah, but the problem with that is the camera adjusts, of course, the brightness. That's, of course, a problem. Yeah, the exposure time is adjusted by the camera, and this means that we're not able to see it quite as nicely because the camera maybe like this. Yeah. See, every time when it becomes darker, then the camera will readjust the exposure time. Yeah. But this gives you maybe a very, very small hint. Yeah. And ideally, of course, the background should be completely dark. But you see that the starch grains, they start to shine a little bit brightly. Yeah. So it's actually a very easy thing. So some people always ask me, what can I do to upgrade my microscope? And they want to buy new objectives and new whatever. And I say, no, sometimes not necessary. Just get yourself a set of polarizing filters. Yeah. And another thing I just want to say is the following. Where is the desk view again? There are these 3D glasses that you can might be able to use, but the problem is that these are circular polarizing filters, and sometimes they do not, when you turn them, they do not completely cancel the light, so you have to find the correct orientation by turning some of them around. Yeah, see, maybe you can look at the temperature here, right? Yeah, and now you turn it around, and now yeah, that's the way it should be. But if you flip one around over here, then you see it will not be completely dark. Yeah, so you're still able to see the degrees. Yeah? So that is yeah. Over here, look, I'm just going to, I don't know. The problem is that with the cover glass on top, the water doesn't evaporate quite as easily. So what I'm going to do now is I'm going to try to put it under the microscope in any case. Let's cool it a little bit because it's kind of hot. Okay. And then let's see if, and then, ha! We love you. So beautiful. I know what to do today. Okay. Yeah. So just make sure you can buy on Amazon. You can buy linear polarizing filters. These are kind of sheets, and you can cut them into any shape that you want. You only need one of these sheets, and then yeah, essentially you can do quite a lot of. I've made a whole bunch of videos already with concerning also looking at crystals and so on. Yeah. Yeah, so that's vitamin C. Oh, look, it's growing. Haha, look, isn't this nice? The vitamin C is growing outwards. Okay. This is a little bit off topic, yeah, but I think it also looks nice. Yeah, so that is something, yeah. See, here we're at the end already. Yeah, they are on Amazon too, but they are at least 4 times the price on average for the same thing. Ah, yeah. Yeah, of course, it's, yeah. So that's basically vitamin C. And on the other side, so that's where the cover glass starts. And I'm a little bit, it would be is a pity that I'm not able to see. So we're now here inside the cover glass. It did not crystallize inside the cover glass yet. Look, it's trying to make crystals over here, inwards. Oh my gosh, could this be? I cannot believe it. I cannot believe it. How many of you actually realized what happened? How many of you realized what happened? I love it. Actually, this is a pretty good demonstration. Do you see those circles? What do you think they are? Yeah? What do you think? Yeah? What is, yeah? What do you think these circles are here? I love it. Yeah, these are the starch grains. Okay? These circles that you see here in the background, these are the starch grains because it didn't disappear, but I tried to dry the slide on my coffee warmer. To make the starch crystal, to make the vitamin C crystals form, and those are now the amorphous starch grains. So I cooked them accidentally. Yeah, so these circles that you see here, these are the starch grains. Yeah, and because 65 degrees centigrade, and I cooked them. Yeah, so here these are the starch grains, and the iodine disappeared because when you heat it, the iodine disappears. Yeah, so. That's basically, that's pretty cool. Let's, yeah. Yeah, that's in bright field. It's more difficult to see. You know what I'm going to try, just for the fun of it? I'm going to try phase contrast. Let's try phase contrast. Does this work with phase contrast? Yup, phase contrast, 40 times. Here they are. Here are your starch grains, the boiled ones, the cooked ones. Yeah. So again, to summarize, the reason why they changed their shape is because I put the slide on my coffee warmer to dry it faster so that we can see the vitamin C faster. But this basically happened as a byproduct. I boiled the starch, and now the starch grains are not nice and oval anymore, but you can see them much larger. Yeah, yeah. So basically, right now, what you see here now is, yeah, it looks melted, as you say. Yeah. Yeah, so, you know what I'm gonna do, of course, now. I know I've got the questions here, and I'm gonna do the questions as well, but I'm just going to try the following, just for the fun of it. Desk view. See, this 65 was a little too much. Let's turn it off. So, where is my iodine? I wonder if it's able to actually go in. Oh yeah. Let's see. Maybe over here a little bit. Yeah. So I'm going to remove again the excess iodine here. There seems to be a little small reaction happening over there. So let's have a look. And nope, I have to remove that filter. Let's see if there is. Yep, see? Here they're starting to stain dark again. Yeah. Yeah, you see, I just added again a little bit of iodine, so this confirms that indeed these large amorphous structures are indeed starch. Yeah, boiled by the coffee warmer. Okay. Yeah. So, okay, what I'm gonna do now is I'm going to quickly have a look at some of your, because it's actually already 1.5 hours, but maybe there are a couple of questions that yeah, I did not answer yet. So let's have a look at those. So I'm gonna do a pretty quick run through here. Okay. Yeah, that is about the carpet beetle. The larvae have interesting hair. Yes. Are you sure that the banana is not moldy? No, it's not moldy. Okay, it's just yeah, has just changed its color. But if it were moldy, then actually you would actually see it. It will look quite differently. Yeah. So yeah, so over here the beetle, the carpet beetle, again. The leaf of I don't know. Grünlilie. Sorry, don't know that. Okay. Thanks for your advices. I bought a 60x objective instead of 100x for observing mold. Works great. Yeah, I like I don't like the oil immersion objectives as much because it's kind of messy, right? So for this reason, I generally recommend, if you want to go high magnification, 60 times, and that generally is most cases good enough. Also because you end up using it more often. You don't have to always clean the objective after every use because of the immersion oil. Yeah, so that's also one of the reasons why I kind of like the 60 times. Yeah. Do any other fruits and vegetables have starch too? I've never thought about that. Yeah, sure. A lot of in plant leaves you can find also quite a bit of starch. Yeah, in some of them. Yeah. If it were to backlight the petri dish and use polarizing filters, would we be able to see the light them as tiny bright specks? Okay, I didn't see that question yet. So the question is a little bit, I don't have, unfortunately, I don't have now potatoes available. Okay, but the question is if I actually did try it now with one of the slides, and the problem is that the camera always adjusted the exposure time, so I was not able to see this properly. This was the problem with the camera. Camera problem was solved. So I talked about how the plant breaks down the starch. It produces enzymes that break it down. Okay. Giant apple-sized grain of starch. Yeah. So again, a camera issue here. I just can skip over this. From research, starch is linked in long chains with small amount of branching. Yes. Glycogen is linked in many branching chains, and chitin and cellulose form single chains that pack together. Yes, that's correct. As a matter of fact, when I teach biochemistry in school, my students have to know the structure and the structural differences between starch and cellulose. Can starch be heat fixed? I don't know about that. I didn't try it now, but what I read somewhere is that heat fixing works with cells because there is protein on the surface, and the protein makes crosslinks with the slide. So starch being a carbohydrate, I don't know if it does this. If we try the slide, will it form crystals? We tried that, and because of the heat, it actually boiled starch. The vitamin C crystals are awesome. Of the 10 or so chemical crystal slides I've made, vitamin C has been the best. Yes. If you want to try vitamin C as well, you can also try to dissolve a little bit of vitamin C in alcohol. It dries faster and the crystals look different. But why do you need to heat the amylase? You don't need heat to let amylase break it down. Okay, so, I think I see what you mean. Generally, when you boil the starch first, because it loses the crystalline structure and becomes softer, then the amylase, the enzyme, can attack the starch better. So in that sense, I also found the hard way that pure vitamin C will scorch and burn before it melts. Yes. It will also oxidize. It will actually turn brown as well in the presence of air. Which temperature did you use with saliva digestion? I had 65 degrees Celsius, centigrade. It was evidently a little bit too high. So the phloem, this was in the banana. We should be able to see it, but might be a little bit difficult to identify. The xylem is easier to see because of its spiral-shaped strengthening structures. Okay. I have not finished the slide vacuum chamber yet. Well, actually, I did make one. But it was not quite airtight. So still working on it. So why the starch has different shapes, I don't know. That doesn't necessarily. Sometimes it doesn't always have to be a reason for something. As long as it works as a storage substance, it should be fine. Thank you for the food syringe tip. And bananas lose starch as they ripen. I talked about this. I tried to make a vacuum chamber using a cup with a hold on the bottom. Okay. Using a vacuum cleaner. Oh, okay. So you try to basically remove the air using a vacuum cleaner. Okay. So very last questions here. Sometimes you might not be able to see mold because even though it's there, it might be too small to see. That is generally correct. That's correct. So sometimes food might look fine, but it's moldy. Is it fine to consume them? Okay, so that's a good point. It is correct that there's sometimes the so-called microcolonies present, and which basically makes the food bad, right? Generally, however, if colonies start to form of mold, then they will grow very quickly once they're there, right? But experience has shown that just because the banana changes the color here does not automatically make it bad. Look, the inside here, is still quite okay. So also those discolorations here is not necessarily mold. And another thing is you know that something is spoiled if indeed if it starts not only to change its color or texture, but it also starts to smell and taste bad. So that's also usually something. But this banana is perfectly fine. I'll just eat it. So this was a slightly stressful live stream for me. Specifically because of the camera situation, but I'm glad that ultimately the things that I wanted to show you, I was able to show. Okay. What I'm going to do now is I'm going to call it quits for the day, because at the place where I am in Central Europe, it's 11 o'clock at night already, but for some of you it might be even less convenient time. Okay, I'm just gonna say goodbye. Hope to see you again next week. I still have to think of a topic for next week, and I hope that I'll also get the camera situation fixed. Okay, all the best. Happy microbe hunting as always, and see you around. And thank you that you have joined me again. Bye bye.